COLDA for giving me the opportunity to share my work. My name is Basetsana Katlo Phakedi, and I'm a Sante Fellow based at Botswana Harvard Health Partnership and a student at University of Botswana. So my project is titled Detection of Hepatitis B virus in cerebrospinal fluid of people living with HIV-associated cryptococcal meningitis in Botswana. So hepatitis B persistence in the central nervous system may bring about compartmentalization where distinct viral quasi-species may occur that harbor escape or drug-resistant mutations, which may complicate treatment. So HBV compartmentalization has not been studied in Botswana, and looking at extrahepatic sites may help us understand HBV pathogenesis and improve HBV treatment strategies. So the aim of my study is to investigate the presence of HBV in the cerebrospinal fluid of people living with HIV, and I had three main objectives, which is to detect surface antigen and HBV DNA in the plasma and CSF compartment. The second objective is to compare HBV detection rates between plasma and CSF pairs. And the third objective is to assess genotypic and mutational diversities among the plasma and CSF pairs. So this was a retrospective cross-sectional study that used residual samples from the Ambition CM study, which enrolled people living with HIV with cryptococcal meningitis aged 18 years and above. So my study started by screening surface antigen and HBV DNA in plasma and CSF pairs, and we went all the way to amplify and sequence the 415 base pair surface polymerase HBV region using the next generation sequencing method for mutational and genotypic analysis. So here I just want to highlight that our cohort were severely immunocompromised with records of the median CD4 T cell counts of 29 cells per cubic millimeters, which means they had advanced HIV. So it is to note that not all participants had plasma CSF paired samples due to shortage of residual samples, so the prevalence is reported per compartment. So when we look at the results, the prevalence of surface antigen in plasma was recorded at 29.2%, and in CSF it was recorded at 18.8%. When we look at OBI, occult hepatitis B cases, which is surface antigen negative but HBV DNA positive samples, we see the prevalence at 35.5% in plasma and 2.4% in the CSF. So when we look at samples that did pair and compared the detection rates, we observed a statistically higher detection rate in plasma than in CSF, as well as HBV viral loads levels were higher in plasma than in CSF. So looking at the genotypic analysis, we managed to sequence 10 samples, of which we derived 3 plasma CSF pairs, and we saw a genotypic concordance where pair 1 and pair 3 had genotype A and pair 2 had genotype D. These results were confirmed by Genotofino. We then moved on to mutational analysis, where we observed mutational discordance. So pair 1 had a higher frequency of mutations in plasma than in CSF. Pair 2 had higher mutational frequencies in CSF than in plasma. And then pair 3 had mutational concordance. And then when we look at compartment-specific, we also observed compartment-specific mutations, where mutations are present in one compartment rather than the other compartment. And these mutations conferred to immune escape and potential drug resistance. So in conclusion, HBV can be detected in the in the central nervous system, and the difference in mutational profiles just show us that the central nervous system may be a reservoir for HBV replication. And then my findings also highlight the importance of looking at extrahepatic sites in HBV pathogenesis, and also call for further studies to investigate the importance of including of the existence of HBV in the cerebrospinal fluid or the central nervous system. So I'd like to thank everyone who was involved in making the study possible, more especially the Ambition CM participants and COLDA and the COLDA Travel Scholarship for giving me the opportunity. Thank you.